Hey Year 10s, just a really quick short tutorial on how to use this codon table and fill out the worksheet. So the worksheet, practicing DNA transcription and translation. It'll also move on to the protein synthesis worksheet. Okay, it is on the back of the one that you've done previously as a summary. Okay. So, really good point here. A codon chart can only be used for decoding a strand of mRNA. I'm just going to put this one over the top because it's just a bit larger so it's easier to see. You can use this one or the other one, it doesn't matter. I'm going to ask for a little assistant. So we're going to get Billy the Bilby. He's going to make sure I don't make any mistakes. Okay, so remember your DNA strand cannot leave the nucleus. The only way it can leave the nucleus is to produce what's called a messenger RNA strand or an RNA strand. Remember that T gets converted to a U on the messenger RNA strand. The messenger RNA strand can then leave the nucleus and head to the ribosome. So we've just done transcription. Once it hits the ribosome out in the cytoplasm, it's going to go through translation. Basically, the end result is producing a protein. Why do we need to produce proteins? Proteins actually keep our body functioning. It's pretty much all about the proteins. So let's have a look. The first one, you can see that there's the DNA strand. They've split them into the triplets, making it a bit easier for us. Okay, so these are the bases on the DNA strand. So what we need to do first is we need to write down the complementary base pair for the messenger RNA, keeping in mind that T will now be uracil. So the first one's TAC, so your messenger RNA is going to be A. Now A bonds with T, but remember it's on the RNA, so it's going to be U for uracil, and you got C and you got G. You're just going to keep going along on that base sequence. I'll do the next one. You've got a G on the DNA, so you're going to have a C. You've got a C on the DNA, so you're going to have a G there. And you've got a G on the DNA, so you're going to have a C. I'll do one more. Okay, so I've done those first three for you. Now we need to figure out the corresponding amino acid. This codon sheet, it's exactly what it says. It's a codon sheet. So you're reading off the messenger RNA. Keep that in mind when you start working on that second worksheet. Okay? All right, so my amino acids of my messenger RNA, there's an A, so the first base is an A. I'm looking in this row here. The second base, so it's the second base here, is a U. So I'm looking in this row, this column. So I'm actually looking in this box here. So it's either going to be isoleucine or methionine. The last one is G. Now if we come over here, our third base, our G is down the bottom. So remember we're in this square because the first one was A and then it was U. Now it's G. It's going to be methionine. Now methionine is actually a start codon. All right. So what you'd then do is you can write your methionine in there. Now a lot of the time with amino acids, they do abbreviate them to the first three letters. So if it's easier for you to do that, so I'm going to have M-E-T, met, okay? And that's my start codon. That's telling the messenger RNA to start the process of making the proteins. So the next one, let's have a look. I might just try and zoom in a little bit for you guys. Okay, so my next one, I've got CGC. So first base is C, I'm in this row. Second base is G, I'm in this column. Oh, look at that. Doesn't really matter what the third base is because they're all the same. However, the third base is C, so I'd come across and we've got arginine. So you can just write in A, R, G. And you keep going like that, right? And then that's how you make your protein. Now, it does get a little confusing. The next two examples, they want you to do the DNA is already there. The mRNA, okay, so you've got to do complementary pair. Remember, it's RNA, so it has uracil. Then you got your tRNA. Remember, that's your anticodon. So you got your codon, which is your messenger RNA, and you got your tRNA, which is your anticodon, which is a complementary of the messenger RNA. Remember, it's still RNA. Once you've filled that out, what's really common is students get down to the amino acid here and they start reading. They use this chart, but they're reading their transfer RNA. Okay. Remember, this table, this codon table, is only for the codon, so it will only work with the messenger RNA. So when you go to do your amino acid for this question, make sure that you're looking at your messenger RNA codons. Okay, third one, there's just blanks. You fill in the blanks. You should be fine with that. Now this sheet as well, I'm asking for this sheet to be done. You've got your DNA strand. You would write your complementary DNA. Complementary DNA. Then it asks you to write down your messenger RNA. Messenger RNA, make it the complement of this one. So basically it's going to be this strand, but your T's are going to be U's. From there, you can do your complementary transfer RNA. But remember, with the amino acid, don't read this sequence, which is your anticodon. You need to read your codon sequence because it's a codon table. A couple of little questions there for you to answer. And then you've got some questions here on the back to answer. All right, I better go. I've got supervision. Sorry, guys. Hopefully that helps you out. Thanks, Billy, for keeping an eye on me. Bye.